My name is Phuong Dao and I'm a PhD student from the School of Computing Science, Simon Fraser University. So today my talk is about predicting chemotherapy response using the optimally discriminated subnetwork markers. So in the conventional approach for cancer therapy is to diagnose patients based on their histopathology profiles and to give the patient with similar diagnosis the same kind of treatment. However, this is a problem in practice because the differences in the molecular profile of this individual, such as the mutation in the drug-resistant pathway, could affect the drug efficacy. So in the modern approach, also the key idea behind personalized medicine, at first we identify the molecular features or markers that make some individuals resistant to the drugs, but not the others. So when a new individual comes, we profile this individual using the high throughput technologies, and we can give the best treatment that's suitable for the individual. So this leads to the this is the motivate the better approach for identify molecular features or markers that could predict chemotherapy response well. So previous approaches in predicting chemotherapy response use the approaches in which we term here single gene based marker approaches. So each marker has a one single gene, and each gene is ranked based on the p-value derived from t-test variance. Such approaches show some certain success in predict drug response in breast cancer. However, we know that the genes usually act together, so it might be possible that the single gene based marker approaches will miss the collective effects of multiple weaker discriminating signals. So alternatively, recent study have shown that these new kind of markers based on network, which we term here subnetwork markers, could capture this deregulation of multiple interacting protein. So it subnetwork marker here is connected subwrap in protein-protein or PPI network. Chan et al. Introduced this single gene marker concept in 2007 and have shown that using subnetwork marker we can outperform single gene marker in the following aspect. We had better classification performance. We have greater overlap among the signature derived from different cohorts of patients, and at last we can pinpoint more about the deregulated pathway of the disease. So here I provide you a better outlines of the method. So given you the expression matrix of the sample, here we have two kinds of sample, from tumor that resistant to the drugs and tumor which sensitive to the drugs, and a PPI network. So at first we map the gene expression to the PPI network, and we only retain the gene from the good map to the PPI network. And then we look in the PPI network a connected subgraph who the gene expression correlate with this phenotype, and then we select the top subnetwork as predictors. So Chuan et al. Introduced the method in the concept of subnetwork marker in 2007, and the authors use a grid heuristic to find these markers, so do not guarantee optimal solution. and presumably optimal solution could yield a better performance. So subsequent study do provide optimal solution but do not provide efficient running time, such as the approach introduced by Chowdhury et al. In RICOM last year, which search for like a pattern of up or down regulated gene that highly associated with the phenotype. Also, another approach introduced by our group last year at ECCB 2010 that we yield a functional module or dense subgraph in this functional association network and some markers. But we use exotic search, so we do not guarantee the efficient running time. So here we introduce the optimally discriminated subnetwork markers, which we term OptDis. The benefit is we can guarantee an efficient running time when the maximum size of the subnetwork is in the order logarithmic of the size of the PBI network. So here I outline the idea behind our approach. So imagine that each sample is a point in high dimension. Suppose you could map 20,000 genes into the PBI network, then you have 20,000 dimensions here. And basically we just perform dimension reduction. So we project each sample here into k-dimensional space. Here k the maximum size of the subnetwork that we would like to look for, such that the sample from the same classes are close to each other, while the sample from different classes are further to each other. So it could be written as the summation of the distances between samples from different classes minus the distances of the sample from the same classes, and we would like to maximize this discriminated score. So however, this is an NP-hard problem. By a reduction from connected K subgraph problem, variation of this problem had been used to detect the differentially expressed pathway, so that in the study by Chiu et al. 2009 and Dietrich et al. 2008. So both studies use integer linear program to model the problem. However, Chiu et al. Show the relaxed version, so does not guarantee the optimal solution, while Dietrich et al. Using branch and bound method, so also does not guarantee efficient running time. So we provide an efficient method to find the optimal discriminated subnetwork markers based on color coding framework, which introduced by Along et al. In 1995. So this framework consists of many iterations. At each iteration, we do the following. So we assign each vertex uniformly at random with one of the K colors. And then we only need to search for the colorful optimal discriminated subgraph, where what I mean optimal here is defined by the maximum distance score, which defined earlier. And what I mean colorful here is the color of the vertices in the subgraph are distinct. So with sufficient number of iterations, it can be proven that we can extract this optimal subgraph with high constant probability. So I should note that color coding approach is the fastest approach that could reduce the running time significantly compared to the naive approach, which we search for only connected subgraph, and it does it by restricting the space to only colorful connected subgraph, even though we need some number of iterations, but it still reduce the running time significantly compared to another approach in the literature. So from now on, I will present the analysis of our method on one of the breast cancer dataset. So we took a dataset from microarray quality consortium study, consists of 230 breast cancer tumor, and this baseline profile before the treatment, and this is a combinatorial treatment consists of 4 drugs. And what we define response here, a tumor response to the treatment is we don't find any cancer in the breast or lymph node, otherwise it's called resistant to the drug. So I outline the analysis that we're going to do. So at first we look at the performance of our method compared to other methods, and then we check out the overlap of our signature obtained from different cohorts of patients, and finally we do the functional analysis of the obtained subnetwork markers. So the dataset comes with 2 different cohorts, consists of 130 and 100 tumors. So the method we going to compare is our method OptDis, the original method introduced by Chuan. We did not read the MI. The functional module introduced by our group for functional association network, we did not advance, and the symbol single gene marker run by t-test, SGM, and the model from this MAQC study. We did not add MAQC. So the workflow as follows. We take one of the cohorts at training cohorts, and then we use different methods to extract these gene markers, and we rank them by the discriminating score. So for single gene, we rank by the p-value of the t-test. And for the subnetwork marker method, for each vertex, we find the optimal subnetwork contain that vertex, and then we rank by their own discriminating score. So we iterate from the top 1 to top 50 gene subnetwork markers, and then we apply k-nearest neighbor classifier on these other cohorts with the not validation cohort. So the analysis that we done on 100 tumor to 100 tumor, we will denote at forward analysis FXD, the other way backward analysis BXD. So finally, the performance is measured by Matthew correlation coefficient. So Matthew correlation coefficient just simply the Bayesian correlation between the predicted vector labels and the observed vector. So if it's equal to 1, it is perfect prediction, 0 is random prediction, and -1 is inverse prediction. So now we look at the classification performance of various methods. So here the x-axis denotes the number of markers, the y-axis denotes the classification performance. So the higher the better. So here one marker here equal to one subnetwork from OptDis, dense and grid DMI, and equal to one gene in the single gene marker model. So we could see that starting from around 16 markers, our method is outperform other methods. So we confirm the improved performance of our method. Also, if we take the average performance using top 1 to top 50 markers. So here we compare our method with also MAQC model. So I will talk more about MAQC. So there are more than 30 groups participate in the MAQC study. So each group nominate a couple models for its analysis. So here we take the best model from each group and the average performance here is the average of the best model. So the white bar here denote the average performance in this forward analysis, while the blue one is in the backward analysis, and the green one is the overall average. So our method show the relative performance in relative performance around 20%. So it might be possible that the improved performance of our method over single gene marker is due to the fact that in our subnetwork we have several genes, and in the previous analysis we only compare one subnetwork with one gene. So now we do the analysis in which we mark the number of genes in the subnetwork. So now its marker here is equivalent to one subnetwork, and it equivalent to Y genes for the single gene marker model, where Y is the number of genes in the subnetworks. And we also compare this single gene marker in which all the genes are from our subnetworks. So in which we denote the SGM of this. So it can be seen here, our method of this is still perform better than the gene run by t-test, but also another important factor is the gene identified using our subnetwork, even though do not have high ranks in the t-test, but still perform better than the top gene run by t-test. So now we look at the overlap of the signatures among different cohorts of patients. So again, here the axis denote the number of markers. So it can be seen that we have a greater overlap of the signature among different cohorts. However, you can see that with only a few markers, we already outperform like single gene marker model. So now, finally, we look at this pathway analysis of the gene derived from our subnetwork markers. So here, the y-axis is the corrected p-value, and this orange line, the dotted orange line, is the significant threshold. So, so here we take the top 5 pathway enriched by the gene from our subnetworks, and more interestingly, the last 2 pathway, IGF and the BI3K signaling pathway, had been known to be drug resistant to more than one drug in the combination, and it has been known that the ER plus tumor are more responsive to the treatment, to the combination treatment. So the first 3 pathways for us are more subtype specific, especially in ER plus tumor. So in conclusion, we just provided you efficient randomized algorithm that could find the optimal subnetwork markers, and we confirm that with the optimal subnetwork marker we can perform better than the one derived using heuristic. And we also confirm again the benefit of using the subnetwork marker over single gene markers in a very difficult clinical diagnostic problem, such as like predicting chemotherapy response. So just a couple of words about our current work. So we know that the growth response are more specific to subtype. So it could be interesting that maybe we could identify the subnetwork marker specific to subtype. So it may provide us a better performance. So in this study, I confirmed the improved performance of subnetwork marker over the single gene marker. However, where do these improved performance come from? So is it merely the collective effect of multiple weaker discriminated genes, or the fact that we use network topology here, like we use connected subgraph in PBI network, functional module in functional association network, or non-pathway or protein complexes? So this is under our current investigation. So that's it. So at first I would like to thank my supervisor, Dr. Sainap and Dr. Esther from Simon Fraser University. I would like to thank to Kendrick Wan, also the co-first author in the paper, Dr. Labak and Dr. Collins from Vancouver Prostate Center. For the funding source of my study, the BEAM Mathematical Training Program and BCID projects. And finally, the Travel Fellowship awarded by ISCB. Thanks a lot.